A spliceosome is a ribonucleoprotein complex comprised of multiple proteins and 5 small nuclear RNAs, abbreviated as snRNA, that together regulate the process of mRNA splicing, also known as intron removal. There are 2 main types of spliceosomes that splice introns out of mRNA. These are called the major and the minor spliceosomes. The major spliceosome handles the majority of the splicing in RNA. Specifically, the major spliceosome recognizes these mRNA sequences at the junctions between exons and introns that are called the 5' and 3' splice sites. In the major spliceosome, the snRNAs U1 and U6 recognize the 5' splice site. The snRNA U5 interacts with the 3' and the 5' splice sites, and the snRNA U2 recognizes the branch point sequence, or the BPS. The BPS is the sequence close to the 3' end of the intron sequence, where a special adenosine residue resides. When the intron is cleaved, this adenosine residue interacts with the 5' guanosine residue at the 5' end of the intron. The minor spliceosome removes minor class introns. Minor class introns, also known as the U12 introns, are defined by their divergent 5' splice site at BPS. The minor spliceosome differs from the major spliceosome in its composition. The minor spliceosome contains 5 different snRNAs. 4 of them, U11, U12, U4 attack, and U6 attack, are unique, and the 5th snRNA, U5, is shared with the major spliceosome. The 4 unique snRNAs are functionally equivalent to the major spliceosomal components U1, U2, U4, and U6 respectively. The secondary structures of individual snRNAs are very similar, and so are their higher-order small nuclear ribonucleoprotein complexes, or snRNPs. For example, U4, U6, and U5 form a tri-snRNP, and likewise, U4 attack, U6 attack, and U5 associate into a tri-snRNP. On the other hand, there are also some differences between the 2 versions of the spliceosomes. For example, in the minor spliceosome, U11 and U12 associate into a di-snRNP before interacting with pre-mRNA, whereas their major spliceosomal counterparts, U1 and U2, do not form a complex and recognize the 5' splice site and BPS respectively, separately and independently. The protein compositions between the 2 types of spliceosomes are near identical. However, several proteins are unique to the minor spliceosome. Both major and minor spliceosomes are essential for life, meaning that an organism will die if these machineries are mutated. While major type introns are found in all eukaryotes, minor class introns primarily appear in the genomes of plants, insects, and vertebrates, but are lost in some fungi, protists, nematodes, and crustaceans. Interestingly, the minor spliceosome is known to be slower to splice than the major spliceosome. The reason why is still unknown. There's a lot to be learned about the process of intron splicing.